This video is about the first part of chapter 2, focusing on heredity, or how our nature, our genetics, and our biology affects who we are. So first, I usually do an activity where I ask you to rate how much nature or nurture influences various traits such as hyperactivity, aggression, obesity, intelligence, athletic abilities, and age of death. So on a scale of 100% nature, your biology and genetics, to 100% nurture, environment, and experiences, or maybe somewhere in the middle, where do you think those things lie? What research has found is that for hyperactivity and aggression, it is both, but a little more genetics playing a role. For obesity, it's pretty evenly in the middle, with both our genes as well as our environment playing a role. For intelligence, it's roughly 2/3 our genetics and 1/3 our environment. For things like athletic abilities, it is a bit more your genes, your nature, than your nurture, and particularly your biological mother's genes that has the most influence there. And then finally, age of death. And like all of these, it is both, but a bit more your genes. So the reason why I like to start with this is that many people overestimate how much nurture plays a role in various aspects of our lives and behavior or personality. And what the first part of this chapter is really focusing on is that genetic aspect, because it is a very important aspect. Not that, not that nurture isn't, but that our genetics are often underestimated in how much of a role they play. So in this chapter, we're going to talk about genes, mitosis, meiosis, genotype, phenotype, genetic disorders, how we inherit different genes. We'll talk about genetic counseling and get into things like behavioral genetics versus genotype environment correlations and interactions, and epigenetics. So let's start with some basics. The nucleus of each cell in the human body, except for our red blood cells and our gametes or our sex cells, but all the other ones contain 23 pairs of chromosomes. So within our cell, this is showing the nucleus, and then within that nucleus or control center of the cell are our chromosomes. Chromosomes are these rod-like structures that store and transmit genetic information. They're made up of DNA and proteins packed into these cylindrical structures. So DNA is deoxyribonucleic acid. It's a double-stranded molecule. It is made up of base pairs. When we talk about genes, that is referring to segments of DNA. And these segments can be hundreds to thousands of these base pairs, or they look kind of like ladder rungs, if you will. We have an estimated 20,500 genes in our DNA. So genes accomplish their task by making proteins, and we share some of our DNA with the simplest organisms, such as bacteria, and then with other organisms that we're a lot more closely related to, we share quite a bit. So with chimpanzees, for example, we're sharing 98 to 99% of our DNA, as opposed to a banana, which we share only 50%. So mitosis and meiosis. Mitosis is the process by which our DNA duplicates itself. So how All of our cells, except for our gametes, uh, reproduce in this way. And you don't need to know like all the phases. But what I'm really wanting you to know is that for mitosis, you'll notice with the parent cell, um, the chromosomes will then duplicate themselves, then they will align and split into two daughter cells. And you'll notice both of those daughter cells have the same number of chromosomes as the parent cell. So it's literally two exact copies of that parent cell. Meiosis, on the other hand, is the process by which our gametes or our sex cells are formed. So referring to ova or egg cells and sperm. So with meiosis, you'll notice the parent cell is there. The chromosomes duplicate themselves similar to the mitosis process, but we see already a little difference here. So after the chromosomes duplicate themselves, you'll notice that similar chromosomes will line up and will cross over and exchange some of their genetic material. And on the slide here, you can see that illustrated by different colored bits of chromosomes being swapped. You'll see like that long blue chromosome now has a little bit of red on it, and one of the long red chromosomes has a little bit of blue on it. The chromosomes then align. They line up a bit differently than they do with mitosis, but they line up all the same. And then that cell splits, and then those two cells split again. And so with meiosis, we end up with 4 daughter cells from that original parent cell. And each of those daughter cells has only half the number of chromosomes as that parent cell. You'll also notice that those chromosomes vary from that crossing over. Crossing over is very helpful because it improves genetic diversity. The better the odds that some of your offspring will have favorable traits that will help them to survive. So with these cells having only half of the chromosomes, they have 23 chromosomes, not 23 pairs of chromosomes, 23 single chromosomes in typical development. The reason for this is because when an ovum, a egg cell, and a sperm cell merge at fertilization, that is when you will get your full set of chromosomes. So this slide shows a karyotype chart, so showing an example of a full set of 23 chromosome pairs. The autosomes, basically the non-sex chromosomes, those are the typical pairs, I'll say. So 22 matching pairs. The sex chromosomes, they may match. So if you are a biological female and you have 2 X chromosomes, if you are biological male, you'll have a mismatch for those sex chromosomes, having an X and a Y. And you'll notice that Y chromosome is much shorter than the X chromosome. And I'll talk a little bit more about why that's significant later. But for the most part, the rest of them match. And if you're curious about learning more about like what genes are on each of these chromosomes, you can follow the link in that title, and that will take you to the Human Genome Project archive that has individual chromosomes mapped out. Pretty interesting. Allele. So for our genes, we have different forms. And I should change that, not 2 forms. Some of them are 2, some of them, there are more than 2 forms of a particular gene. We end up getting 2 genes, one inherited from each parent. Our alleles will be located on both chromosomes in a pair in the same place. So they do kind of line up, if you will. our alleles can be homozygous, meaning that both are alike, or they can be heterozygous, meaning that the alleles differ. So an example I like to use if we talk about dark hair, brunette hair. Brunette hair is brunette hair. You might get 2 copies of brunette hair, one from, again, each of your biological parents. You could get 2 blonde hair alleles, again, one from each parent. Both of those examples are homozygous, where you are getting 2 of the same allele, or you could inherit 1 brunette hair allele and 1 blonde hair allele. So your alleles are different, and then what trait you actually have will depend on the relationship between those alleles. And I did use, well, we'll talk about that on the next slide. There is a link here in the title to a little TED Ed video that also goes over this. So genotype and phenotype. Genotype referring to an individual's genetic information that we inherit from our biological parents. Phenotype, the directly observable characteristics, which depends on that relationship between the alleles and can also be influenced by our environment as well. So on the previous slide, hitting that slide button, I wrote brunette hair with a capital B, and that was on purpose. So brunette hair is what we call dominant over the recessive gene of blonde hair. And dominant recessive patterns of inheritance basically mean you only need 1 copy of a dominant trait in order for it to be expressed phenotypically. Whereas for recessive traits, you need 2 copies in order for that to be expressed phenotypically. So if I put our possible genotypes here, here are homozygous types. You might get 2 brunette alleles, you might get 2 blonde alleles. In terms of what your phenotype would be, you're going to get brunette hair if you have 2 brunette alleles. You will have blonde hair if you have 2 blonde alleles. If you are heterozygous, your genotype, you have 1 brunette allele and 1 recessive blonde allele. Well, what phenotype will you have? Because brunette hair is dominant over blonde, you will have brunette hair, but will carry the recessive blonde hair allele. So here's our slide, dominant recessive inheritance. Again, dominant allele will determine the phenotype, even if it's paired with a recessive allele. The only way to get phenotypically a recessive trait is to get both of those recessive alleles. And several traits follow this dominant recessive inheritance pattern. For example, brunette hair or dark hair, really, in general, is dominant over blonde hair, is dominant over red hair. Curly hair is dominant over straight hair. Various traits like that follow a dominant recessive pattern. Not everything does, though. Sorry, let me backtrack just a bit. So why do we care about recessive traits. Why does it matter if you're a carrier? We do get concerned with this because there are several serious genetic diseases which are recessive. So things like phenylketonuria or PKU, things like cystic fibrosis, Tay-Sachs disease, sickle cell anemia. Those are recessive traits. So it's very important to know if you're a carrier of a trait like that, because if you have children with someone who is also a carrier, so say draw a Punnett square showing potential outcomes for offspring. If you have let's see, cystic fibrosis, for example. So most people don't. So they're healthy. I'll use capital F for cystic fibrosis, healthy or not having cystic fibrosis. So I'm going to say equals healthy. And then a lowercase f to signify recessive, and that is having cystic fibrosis. So if we talk about somebody who is a carrier, right? So biological parents having heterozygous alleles, heterozygous genotype, right? In terms of their potential offspring, their offspring might inherit the dominant healthy copy from both. They might inherit one of each, dominant healthy copy and a recessive unhealthy copy, or there could be inheriting two recessive copies, in which case that individual would have cystic fibrosis. So we can see there is a 1 in 4 or a 25% chance that their offspring, any child they have, would be healthy with two dominant healthy copies of the gene. There's a 50% chance that their child would be healthy but a carrier of cystic fibrosis like themselves. And there is a 1 in 4 or 25% chance that their child would have cystic fibrosis. So while we're not too concerned about things like the hair color or whether you have curly or straight hair, some of these recessive traits again determine whether you have diseases, serious genetic diseases, and so becomes an important thing to be aware of. So not everything follows, not all traits follow dominant recessive patterns. We have codominance and incomplete dominance, which is when both alleles are expressed in the phenotype. So with codominance, we get a combined trait. An example of this is the AB blood type. So type A blood is dominant over type O. Type B blood is dominant over type O. O is recessive blood type. But A and B, they are not, one is not dominant over the other. And so you can have type AB blood. In incomplete dominance, you get an intermediate between the two traits. An example of this is sickle cell anemia carriers. So if you have two recessive copies of the sickle cell anemia trait, you will have sickle cell anemia, which is a serious disease in which the red blood cells are sickle-shaped or crescent-shaped as opposed to round. Because they're crescent-shaped or sickle-shaped, they catch on one another in our veins. And this causes them to form clogs in the blood vessels to block the flow of blood. It causes intense pain, swelling, and tissue damage because oxygen is not able to get to those tissues. However, for people who are heterozygous, they have a copy of the healthy red blood cell trait and they're a carrier of sickle cell anemia. They're typically protected from having the disease, but when they experience oxygen deprivation, such as at high altitudes or after intense physical exercise, that recessive allele asserts itself and a temporary mild form of the illness occurs. So you get a little bit of that intermediate for that situation. So all of the things I've talked about so far follow what we call monogenic inheritance. And comparatively, monogenic inheritance is pretty far less common than polygenic inheritance. So monogenic, let me write that, monogenic, referring to mono meaning one gene influencing the trait, but many more of our traits are polygenic, poly meaning many or several, like a polygon, right? So polygenic inheritance, many genes affecting a single characteristic. Some examples: height, weight, intelligence, personality, your risk of psychological disorders, things like that. Many traits are, or many genes are affecting one trait. Which is why it can be difficult to study traits, especially when we're talking about how traits are passed down and which genes cause a particular trait, because it could be hundreds. Was it for schizophrenia? They've identified over 100 different genes that influence your likelihood of developing schizophrenia. And it's not 100% genetic. So environment plays a role too. So we also have all those environmental factors. So it can get pretty complicated. So genetic disorders. So I mentioned before, there are the recessive disorders where you must have the homozygous recessive alleles in order to have the disease. Those are things like sickle cell anemia, cystic fibrosis, phenylketonuria, or PKU. There are some genetic disorders which are autosomal dominant disorders. So it is, even if you're heterozygous, you will still have that disorder because it is a dominant trait. So even if you have one, right, dominant means that trait will be expressed phenotypically. And so those are diseases like Huntington's disease, Tourette syndrome, dominant, following a dominant pattern of inheritance. We also have sex-linked disorders, or sometimes referred to as X-linked disorders. And that is when a harmful allele is carried on the X chromosome. So when I called your attention to the fact that the Y chromosome is shorter than the X chromosome, meaning that there are fewer genes on it, this is where this comes into play. So biological males are more at risk of sex-linked disorders because they only have one X chromosome. So they're only receiving one copy of that trait, one allele. And if that allele is an unhealthy copy, then they will have that trait. Females, biological females, on the other hand, have a good chance of having a dominant healthy allele on at least one of their X chromosomes to help counteract if they do get the recessive harmful allele on the other. So a couple examples of these sex-linked disorders: Fragile X syndrome, hemophilia, things like certain types of color blindness. Those are things males are more at risk for because they only have one X. So if I were to draw a little Punnett square here, right, and say we have biological mom and biological dad, that their chromosomes on here. Say, because it's an X-linked disorder, say biological dad has the disorder, so I'll put a little lowercase h here for hemophilia. Biological mom, say she is healthy, but she is a carrier. Or actually, sorry, let's say she has 2 healthy copies. She's not a carrier. In terms of potential children, we could have oops, no, I forgot. She does need to be a carrier. Sorry. Sometimes I mess up my example. Erase. Try this again. Oop, and now I'm on the wrong slide. So let's pretend. I think it's dad needs to be healthy. Mom needs to be a carrier. So again, capital H meaning healthy, the lowercase h denoting a recessive trait, in this case, hemophilia. We have biological mom who is healthy but is a carrier, and biological dad who is healthy in terms of potential offspring. We could have these odds. So there is 1 in 4 chance, 25% chance that the female, they will have a female child and have healthy, 2 healthy dominant copies. They could have a female child who is a carrier, but will be healthy herself. They could have a child, a male child, who is healthy, 1 in 4 chance of that. They also have a 1 in 4 chance so that they will have a male child who is who has hemophilia. And that is again because it is solely determined by that trait on the X chromosome, because there is no corresponding allele on the Y chromosome. So that is why males are more at risk for some of those disorders. Getting into some of our other chromosomal abnormalities, most of the chromosomal abnormalities happen during the meiosis process, when the ovum and the sperm are formed, and the resulting abnormalities often produce disorders with various mental and physical symptoms. We can also have autosomal chromosome disorders or sex chromosome disorders. So the autosomal chromosome disorders, individuals inherit too many or too few chromosomes. An example of this is trisomy 21, also known as Down syndrome. What happens with this is the 21st chromosome pair fails to separate properly during the meiosis process. So either the sperm or the egg will have 2 21st chromosomes, and then the egg or the sperm, the other one will just have 1. When they come together, when the egg is fertilized, the individual ends up with 3 21st chromosomes instead of the typical 2. This is the most common chromosomal disorder, occurring in 1 in 691 live births. It leads to intellectual disability, typically problems with the cardiovascular system, with the heart, shortened lifespan, and certain physical characteristics, facial characteristics, like wide set eyes. So that is caused by having that extra chromosome. For sex chromosome disorders, this is when there's a problem with the extra Y chromosome, either inheriting an extra one or not inheriting an X chromosome. There are various conditions. A lot of the times, puberty will be delayed or will cause the sex characteristics of the opposite sex to develop. Additionally, if it's one of the variations where there's an extra X chromosome or a missing X chromosome, that will cause intellectual deficits. So two of the most common sex chromosome disorders are Turner syndrome and Klinefelter syndrome. With Turner syndrome, it affects cognitive function as well as sexual maturation. The external genitals are female in appearance. The ovaries, however, do not develop. So individuals with Turner syndrome are infertile. They also have a short stature and other physical characteristics. And again, that results when an individual has one X chromosome and that's it, no other sex chromosome. So that's an X and a naught. Klinefelter syndrome, on the other hand, is where an individual inherits two X chromosomes and a Y chromosome. So for these individuals, this causes the testes to be small. Some breast development will occur. These individuals are also infertile, and they experience low levels of testosterone. So again, a lot of the times these sex chromosome disorders not recognized until adolescence, where there is that delay of sexual maturation or the development of different opposite sexual characteristics start to develop. Chromosomal disorders do become more common the older an individual is. So this is looking at Down syndrome specifically for women based on the age at which they give birth. So they're showing the younger than 35 line, a 40 or a 35 to 39-year-old line, and a 40 and older. And they're also showing this over several years, so from 2002 to 2014. So you'll see with people who are younger than 35, the rate of Down syndrome among live births per 10,000 live births is fairly low, and that's been very consistent over time. With those 35 to 39, fairly consistent. You can see the regression line, that's the mean, regression to the mean, showing kind of the average across the years. There is a higher incidence of Down syndrome among those who give birth between 35 and 39. It has remained fairly stable though over that time period. And then for those 40 and older, we see a lot more variability and a slight increase, a bit of an increase in that trend over time. And this is not necessarily that Down syndrome is becoming more common based on anything in the environment. This might just be because more women are waiting to have children. And so if you're, again, 40 and older, you have a higher likelihood of having a child with Down syndrome. So that's probably what is accounting for that increase. Genetic counseling is a medical specialty that helps parents to assess the chances of giving birth to a child with genetic defects or chromosomal abnormalities. Who is it for? It is for individuals who have had difficulties bearing children, or if they know genetic problems run in their family, or if the potential mother is over 35. Again, because that there is a higher risk of genetic disorders or chromosomal abnormalities for women 35 and older. So genetic counselors, they do not tell you you cannot have children. They merely help you determine your risk um, through various questionnaires, medical histories, and genetic tests run on the partners who are wishing to procreate. And this gives potential parents options. So they might choose to adopt or not to have children, or they might choose to use a sperm and or egg donor, things like that, depending on their family goal. So behavioral genetics, this is a field which focuses on determining the contributions of nature, our genetics, our biology, and nurture, our environment and our experiences, to the diversity in human traits and abilities. So there are some limitations to looking at complex characteristics. A lot of the times, these things need to be studied indirectly rather than directly. This is done through kinship studies, which compare the characteristics of family members in order to get what we call a heritability estimate, which is a measure the extent to which individual differences in complex traits in a population is due to genetic factors. So with kinship studies, we can compare twins, or we might compare adoptees to their adopted parents versus their biological parents. Again, trying to determine if something is more based on genetics or on environment. So for example, if an adopted child is more similar to their biological parents on a particular trait, then we'll know it's more influenced by genetics. If instead they're more similar to their adopted parents in that trait, then we can assume that environment and experiences are having more of an influence on that trait. So heritability estimates range from 0 to 1. You can kind of think of them in terms of percentages. So a heritability estimate for intelligence is 0.5. Which means that half of the variation in intelligence person to person is due to genetics. So for example, if I have intelligence scores for child A and intelligence score for child B, the difference here between their scores, 50% of that difference is what we're referring to. So 50% of that difference being due to genetics compared to their environment. So a limitation, heritability estimates may not generalize to a population. Sometimes they are misapplied. They tend to exaggerate the role of heredity. So they're of limited usefulness, but they definitely can help us out. The other the most common type of kinship study compares twins, specifically fraternal twins with identical twins, or sometimes identical twins who are raised in different environments. And the reason for this is because with identical twins, they share 100% of their DNA compared to fraternal twins, which only share roughly 50%. So with identical twins, or what is more technically known as monozygotic twins, you might recognize that mono prefix. Remember, mono means one, monozygotic meaning one zygote. So how identical twins form, or monozygotic twins form, in an ovum, an egg cell is released. It is then fertilized by a sperm cell. That fertilized egg is known as a zygote. So we don't just say one cell. We obviously become trillions and trillions of cells, right? So that one cell during its development, it's dividing, right? Becoming 2 cells, becoming 4 cells, becoming 8 cells. And during that clopping process, sometimes that clump of cells will separate into 2 different clumps of cells. And that is how identical twins form. So they share essentially their entire DNA because they are from that same zygote. So they got the same DNA from biological mom and biological dad, full set of chromosomes. And so they're essentially clones of one another. And they're also sharing that same prenatal environment. With fraternal twins or dizygotic twins, di referring to two. So two zygotes. So sometimes two eggs are released. Those two eggs are each fertilized by a different sperm, and then those zygotes will, divide and replicate and develop into two individuals. These individuals will share roughly half of their DNA, just like you share with any of your full biological siblings, right? And so fraternal actually means brotherly. So how much DNA you share with your brother, if you have one. So with fraternal and identical twins, with kinship studies, we like to compare identical twins with the fraternal twins. If identical twins are more similar on a particular trait, such as intelligence, compared to fraternal twins, then we'll know that genetics has more of an influence on that trait. And by the way, that is accurate. Identical twins are more similar in their intelligence score than fraternal twins, even when both are raised together. And of course, they're all sharing they're each sharing the same prenatal environment as well. So it's not all about genes, of course. So let's talk a little bit about how genes and environment interact. Genotype environment correlations refer to how our genes influence the environments that we're exposed to. And the way this happens typically changes with age. So with passive correlation, this is termed passive, meaning that the child doesn't have much control over it. Typically, the child care providers, the parents, the guardians are providing the environment, and that's typically based on their own heredity, their own experiences, their own preferences. So for example, parents who are good athletes are more likely to expose their children to swim classes or t-ball or things like that. The child may become a good athlete in part from inheriting their parents' genes, but also in part from being exposed to athletics. Evocative correlation, on the other hand, is when children evoke responses influenced by their own heredity. And these responses then strengthen the child's original style. So for example, an active, friendly baby tends to receive more social stimulation than a quieter baby, right? A baby that's cooing and moving around, people are more likely to interact with it, to make sounds back or to talk to the child. And that extra social stimulation tends to increase the social responses that the child gives as well. So evokes more social responses from others, and that strengthens the original style. Active correlation is what happens at older ages. And so this becomes more common as we age because we have more freedom to seek out environments that fit with our genetic tendencies. So our tendency to actively choose environments that complement our heredity is called niche picking. So for example, a musically inclined child might join the school band. They might practice playing an instrument of their choice compared to somebody who's not musically inclined. So we also have genotype environment interaction, which is the idea that because of our genetic makeup, we differ in our responsiveness to qualities of the environment. Or in other words, people have unique genetically influenced reactions to particular experiences. Additionally, similar responses can result from different gene environment combinations. And an example of this is intelligence and how much stimulation you receive in your environment. Remember, intelligence is partly determined by our genes, partly determined by our environment, by our experiences. So we often talk about intelligence in terms of a range, reaction range, in which some individuals, maybe their range is so your genes essentially set your possible range for your intelligence. And then your environment will determine whether you end up at the top end of that range or the bottom end of that range. So if I show, let's draw a little chart here, say one individual, their potential intelligence scores, we'll do intelligence along our y-axis, and we'll do environmental stimulation or enrichment. Along the bottom, low to high there, and then intelligence scores along our Y, and again, low to high. So one individual, their range, potential range of intelligence score could be between here and here, depending on how much stimulation they get from their environment. So they might be, if they're getting less stimulation, they might end up on that lower end of their potential range. Or if they get a lot of environmental stimulation, they'll end up on the higher end of that range. Versus another individual where maybe their reaction range is a lot larger. Their genetics are predisposing them to be far more sensitive to their environment than other individuals. So maybe they have, with a high level of enrichment in their environment, they're going to end up pretty high in intelligence. Versus if they're in a poor environment, they might end up very low in terms of their intelligence. So again, genes kind of setting that range, and then environment determining where you end up within that range. And then last but not least, we have epigenetics. So development takes place through ongoing bidirectional exchanges between heredity and all levels of our environment. Our genes affect our behavior and our experiences and behavior affect how our genes are expressed. So the concept of epigenesis reminds us that development is best understood as a series of complex exchanges between nature and nurture. So certain types of environmental exposures can cause our genes to be turned on or off, thereby altering our genes' expression, altering our phenotype. One way this happens is through methylation. So a methyl group is a chemical marker, and it can attach along your DNA at a particular genetic site, right? A molecule attaching there, and it can turn a gene on or off. Okay. We can also inherit these genetic markers. So if our parents were exposed to something that turned off a gene, we can inherit that gene in its turned off form. So we'll inherit that methyl tag as well. So it's not changing our DNA, but it is changing our trait by turning off or turning on a particular gene. So again, nature and nurture, it is definitely both, and it's an interaction as well. It's not always a clear, it's 100% this or that, but rather a complex mix of the two. Thank you.